Okay, so this is a kind of overview of foraminifera, distal Meiofauna foraminifera. And this opening slide shows two groups of really quite contrasting organisms. So on the left here, we have three very conventional looking calcareous multi-chambered forams. These are the kinds of forams that you typically find in textbooks. And then on the other side here, we have three organisms which I think are also forams, which look completely different. So one of the main points really I want to make in this talk is that the kinds of forams that you will find in the CCZ are dominated really by these unfamiliar and largely undescribed kinds of species. Now, there have been enormous advances in recent years in our understanding of the relationships between different eukaryote groups. So this slide shows some of this wider context. And each of these lines is a lineage, an evolutionary lineage. So three of these, the plants, the fungi, and the animals, these are the organisms we see around us every day, the kinds of things we buy in the supermarket and eat. And but the the vast bulk of the diversity of eukaryotes consists of microorganisms that we don't normally see. So a lot of the diversity is hidden. And the forams sit down here. They're in a group called the Rhizaria, and the foraminifera are unusual amongst eukaryotes or these single-celled eukaryotes in that they span quite a large size range. So some of them you can actually see. So I should say that most of these lineages are protozoans. And so forams are protozoans. So they are a phylum, the phylum Foraminifera, protists or protozoans within the supergroup Rhizaria. And here they're related to the Polycystina and the Acantharia, which until recently were grouped together as radiolarians. The radiolarians are now known to be a polyphyletic group. Anyway, these are the closest relatives, or some of the closest relatives of the forams. So forams occur in all marine environments, from intertidal mudflats down to the deepest part of the oceans, and you also find them in freshwater and even terrestrial environments. As I mentioned before, they span an enormous size range, from the Meiofauna up to the Megafauna. And well, in this talk, I'll be really focusing just on the Meiofaunal size range. Actually, the Megafauna, these giant xenophyophores, which are very common in the CCZ, these were dealt with in the Megafaunal workshop in Williamshaven a few years ago. They are found in soft sediments, and they're also found on hard substrates. So if you look at nodules, the nodules are encrusted with numerous forams or foram-like organisms. Unfortunately, there's not really time to talk about these encrusting faunas, but they're also very diverse and very interesting. Now, foraminifera become an increasingly important component of the Meiofauna with increasing water depth. These are data mainly from the Northeast Atlantic, apart from this, the Arabian Sea. And at abyssal depth, so more than 4,000 meters, forams can make up 60, 70, 75% of the Meiofauna. I notice this is a little bit different from what Pedro said, where Pedro said that the nematodes were the dominant group in the Meiofauna. Well, my claim is that it's the forams which are the dominant group, but anyway, we'll discuss that later. This is just a little bit of a diversion, just a few remarks about methods, because the methods used to study forams are rather different from those used in the case of the metazoan microfauna. So the most commonly used sieve fractions are 150, 125, and 63 microns. The 32 micron sieve fraction, which is the standard lower limit for the metazoan myofauna, is rarely used. We, in the Abyssline project that I've been working on mainly in the CCZ, we've used 150 micron fraction and also a 300 micron fraction for comparison with a macrofauna. The other point is that we stain residues with rose bengal and then we hand sort them under a binocular microscope. And the Ludox flotation methods, which are normally used for metazoans, they don't work very well with foraminifera. A key characteristic of the foraminifera is this complex net-like system of pseudopodia, and these are the means by which the organism interacts with the environment, and so the pseudopodia are involved in all sorts of life processes like reproduction and shell formation and feeding. The other really characteristic feature of foraminifera is the presence of a shell, a testor shell. In fact, there are some naked forams which don't have a test, but these are really quite difficult to see in fixed samples. Well, I guess they're impossible to see in fixed samples. Really the main, or the two main publications, taxonomic publications, are the 1964 Treatise on Invertebrate Paleontology. This is part of a sequence of volumes by Meiofauna and Tappan. So this is a 2-volume work which is devoted mainly to the foraminifera, although it deals with other groups as well, such as the testator medium. And then the 1987 update, which is published again as a 2-volume book. There's also this nice atlas of benthic foraminifera, which is a mix of modern and fossil species, mainly from ocean drilling project samples. So, these are fossilizable species, this is a nice book. It's got lots of nice pictures in it, but most of the forams that you find in this book you won't find in the CCZ. The traditional classification of forams is based on the it's based almost entirely on the test characteristics. For higher taxonomic levels, the number of chambers, whether the test is single-chambered or multi-chambered, is important, and the wall composition. So the 3 main types of wall are organic, agglutinated, and calcareous. And then among the calcareous groups, you define groups based on the structure of the calcareous wall, the way it's layered, and the way that the crystals are arranged, and also its composition, whether it's composed of aragonite or calcite, which are 2 different crystalline forms of calcium carbonate. And then for lower taxonomic levels, things like test shape, chamber arrangement, and the type and location of test apertures all these features are important. Now, these are the main orders with modern representatives based on the work of Loeblich and Tappan, and I just want to sort of run quickly through these main orders. So starting with the single chamber, these 2 single-chambered orders, the Allegromids, which have an organic test wall, and the Asterorhizids, which have an agglutinated wall composed of particles which are picked up off the seafloor and stuck together with organic cement. Now, we now know that these 2 orders are hopelessly polyphyletic. That means that they are not natural groupings. And so it's no longer advisable, really, to recognize these 2 groups as formal taxa. Then the liturolids, these are multi-chambered. The wall is agglutinated, and the cement which holds together the particles of the wall is organic. The test is typically planispiral, so the chambers are arranged in a spiral or in one plane. The tricaminids, multi-chambered, agglutinated wall with an organic cement again, but the chambers are arranged in a proper spiral arrangement. So this is an arrangement of chambers like a snail shell. And then the textularids, these are multi-chambered, agglutinated again, but in this case the wall is, the cement is calcitic, made of calcium carbonate instead of organic material. And these are typically biserial or triserial arrangement of chambers. And then we have a series of calcareous taxa. The meiofauna are usually multi-chambered, although this is a tubular, a tube which is coiled. But all these types are multi-chambered. The wall is composed of high magnesium calcite. There are no pores in the wall, and the wall is opaque. It's often white and shiny. It looks rather like porcelain. The genids are either single-chambered or multi-chambered. You get a lot of these very delicate flask-shaped single-chambered genids in deep-sea sediment. You get a lot of species, but they're never common. The wall is composed of low magnesium calcite, and the crystals are arranged in a single layer without any with pores, and the wall is hyaline or glassy. And then the rotolids. Now this is really the most important group of calcareous forams. Again, these are multi-chambered. Usually the chambers are arranged in a low spiral or planispiral pattern. The wall is composed of low magnesium calcite arranged in two layers with pores, and the wall is hyaline, which really is just a fancy way of saying it's glassy. And finally the rogotinids, which are similar to the rotalids, but the wall is composed of aragonite, which is another form of calcium carbonate rather than calcite. Now, in recent years, there have been, tremendous advances in molecular analyses of forams, and this really has led to a new classification, which Jan Pawlowski published a few years ago, and this classification really recognizes three main groups. The Globothalamia, which includes the multi-chambered agglutinated taxa, and the rotalids, the rogotinids, and the globigerinoids. The globigerinoids I didn't mention. These are planktonic calcareous forams. And then the Tubulothalamia, which includes the meiofauna, and then two small taxa with a tubular spiral, a spiral tubular test. And the spirulinids, the test is calcareous, and the amidicids, the test is agglutinated. And then we have the monophyletes. This is a heterogeneous polyphyletic group of single-chambered taxa. And so you can think of the monophyletes as basal forams. They come at the base of the evolutionary tree. And so through molecular work, mainly done by Jan and his team in Geneva, a system of clades has been recognized. So really all we have is this system of clades which is based on DNA sequences. There's really no formal taxonomy for these organisms. The other point is, in fact, the micropaleontologists do use a morphology-based classification for the monothalamus, but it's really not very much use for the kinds of forams that you'll find in the CCZ. So there is a morphology-based classification, but it's best not to get into that. Some of these clades include a mixture of taxa with organic and agglutinated test walls. And so this really undermines these two orders, the Allegromeida and the Astrorhizida. So they seem to have no phylogenetic significance because they're just mixed up in these clades. At this point I should mention the Sticomata. Sticomata are small pellets of waste material, mainly clay minerals, that are retained within the test or the cell. And these are found in many deep sea monothalamids. So you can see them here as these little, dark particles which are sort of mixed up with the cytoplasm in these monothalamids. This SEM picture shows that they're composed of clay particles. So monothalamids are a dominant group in the abyssal deep sea, and the vast majority of them are undescribed. These are some examples from the Kaplan East site in the eastern part of the CCZ, and all these are undescribed even at the genus level. And here we get into very shaky ground because unfortunately you don't have a whole array of morphological characters as you do with nematodes and harpacticoids and chinorhynchs that you can use to classify these things. You have to really try to recognize species based on the shape, the nature of the agglutinated test wall, whether they have sticomata. This one has sticomata, this one doesn't. The aperture, whether they have an aperture, or whether it's just a simple sphere. So there's not so many taxonomic characters that you can use to differentiate between species. Now, a key point in considering the forams is that much of the CCZ lies close to or below the carbonate compensation depth. So CCD for short. The CCD is the depth in the ocean at which the dissolution of calcium carbonate in the form of calcite exceeds the supply. So carbonate doesn't accumulate. In the North Atlantic, it's located deeper than 5,000 meters. In the Pacific, it's located between 4,000 and 5,000 meters. Generally, it's deepest in this equatorial zone, and then it becomes shallower as you go to the north and south of this equatorial zone. And this Russian data from a paper by Seabed shows that that calcareous forams disappear at around about 4,000 meters in the Pacific, and below about 4,000 meters, you get just simply agglutinated forms. There are no calcareous species. This is because of the CCD. This actually isn't true. Isn't strictly speaking true. You do get calcareous forams in small numbers below the CCD. For example, this paper by Smith, 1973, she worked on these two transects in the North Pacific, and she found small numbers of calcareous forams down to 7,000 meters. So you will find them below the CCD, but they're not common. Some more important papers from the Pacific. This paper by Enger et al., published recently. Station M, which is on the continental margin of California, to the north of the CCZ, and she found that calcareous species made up 10 to 20% to 30% of the fauna. This was at about 4,000 meters. In the central North Pacific, deeper than 5,500 meters, Bernstein found that only 6 out of over 5,000 forams were calcareous, but they were working on the 300 micron fraction, where you wouldn't expect to find many calcareous forams anyway. Then there's this paper by Rezig in the Peru Basin, and she found that calcareous species dominated between 2,600 and 4,100 meters, and below 4,100 meters there are no calcareous species. The base map here is from a 3-volume monograph by Saidova a Russian author. In fact, this is in Russian, this monograph on the benthic fauna of the Pacific Ocean. Most of the samples came from the northwestern Pacific. There's not so many in the CCZ. There are a number of studies on monothalamus forams from the CCZ. From going from the western part to the eastern part. These are concerned with the megafauna, the xenophyophores, the really big forams. Then there's the paper by Kanskaya on the macrofaunal sized forams. But there are three studies which deal with the meiofaunal sized monothalamus forams from the CCZ. These are by Okawara. This is a PhD thesis, which is largely unpublished. And then Nozawa, a study in the eastern Kaplan East site, which is quite close to the UK1 area. And then a paper by Vansujevska et al., and that's in the IOM area. So there are a few studies of meiofaunal forams in the CCZ. This is the Nozawa et al. Paper, Nina Okawara's PhD thesis, and the Vansujevska paper. Diversity. Deep sea faunal communities are typically very diverse, even in food-rich abyssal regions. So this is an example from the Northeast Atlantic, the Porcupine Abyssal Plain site, where the top 1 centimetre, just the top 1 centimetre of this core sample yielded 608 foraminifera belonging to 131 species. Notice that there's a long tail here of rare species, so more than half of the species were represented by just 1 or 2 specimens. It's the same story in the CCZ. These are data from a single sample from the UK1 area, 4,150 metres. Again, this is just the top 1 centimetre. This is 150 micron fraction, and we found 797 specimens and 169 species just in this single sample. And notice again that there's a long tail, well over 50% of rare species. Well, by rare I mean represented by 1 or 2 individuals. Now, how can we classify these monothelmin-dominated CCZ assemblages? This is quite a challenge because really for the monothelmins there's no well-established taxonomy. And in the UK1 area we use a pragmatic mix of informal morphology-based groupings, tubes and spheres and so on, and more formal taxa. So I've divided the groups I've got 3 categories of groups here: familiar taxa, less familiar groups, and mainly unknown. So the familiar taxa include mainly multi-chambered taxa, so rotalids, Meiofauna, Virginids, and so on. And these are coded in green. The less familiar groups include 2 more or less formal taxa, but of uncertain affinity. So the Camochiaceae, which I'll say more later, and these Nodellum-like forms. And then the rest are monothelmids. So these more or less formal taxa are yellow, and the monothelmids are this pinkish colour. And then the mainly unknown groups are mainly monothelmids. So I just want to go through some of these groups to show you what they look like. So first of all, the familiar groups, so these are rotalids, robertinids, miliarids, and multi-chambered agglutinated forms. These have a high fossilization potential. And because of that, they've been widely studied by paleoceanographers and micropaleontologists. So this is why these are well known. Because they're important in the fossil record, they're important for reconstructing ancient oceans and interpreting the paleoecological conditions in the past. So the Hormoselids, these are also familiar. The hormicenids are the simplest of the multi-chamber taxa. They consist of a linear sequence of chambers, just a simple row of chambers. And these have a fairly low fossilization potential, so and as a result, much less is known about them. They're less well studied, but they're common and they're diverse in most deep sea settings. So there's some species which are described, there's a lot of species which are undescribed. And then we come to the less familiar groups, so allogromids, which are organic walled monothelamids with one or two apertures, different shapes, oval shapes, sort of tubular shape, and then the secaminids, which are agglutinated monothelamids with one or two apertures. And I put these in inverted commas because these are just convenient groupings, and they're not to be considered as formal taxa. So these are typical monothelamids. They're mainly undescribed, and these are all from the CCZ. So sometimes you find these secaminids which have shiny surfaces. We call these silver secaminids because they have reflective surfaces. Sometimes they have this species has a big aperture. This species has apertures at either ends. It has two apertures. This one has two apertures as well, although you can't see them, they're very tiny. So they have different numbers of apertures, they have different shapes. Sometimes they have stichometer, sometimes they don't have stichometer, and these are the kinds of characters that you use to differentiate them. And then we have the Camochiacea. So I've color-coded these in yellow because these belong to a more or less formal taxon. So the superfamily Camochiacea, described by Tyndall and Hessler in 1977. They're usually called Camochi for short. And this is a group which is confined to the deep sea, and they're extremely abundant in the abyss. They're mainly macrofaunal in size, but you will find them in the Meiofauna, sometimes as fragments. So there's about 14 described genera. Some of them have chambers, little bead-like chambers. Other forms consist of branching tubules. This one, the tubules divide into tufty extremities. Sometimes you find these mudball-like structures. And these are also Camochiaceans. These are largely undescribed. But there is some taxonomy for this group, and there are described species. Then another less familiar group are these nodellum-like forms. These have a basically organic test, which is usually brownish in colour. Nodellum itself is tubular, so these needle nodellums are very, very fit. We just call them needle nodellums, they don't have a name. And they're just a tube. Other nodellums are wider. And then we have this genus Ratigella. This actually has a name. It was described from the Peru Basin, and this is a kind of a segmented structure. These things are very widely distributed. You often find them in the deep sea. They're usually not very common, but they are occasionally a dominant group. For example, in the Challenger Deep, the deepest point in the ocean, you get lots and lots of these things. And then we have these chain-like morphotypes. Again, I don't know where these fit in really. I mean, are these monothelamids, which are sort of budded new chambers, or are these some kind of, true multi-chambered forms? It's not really clear what these things are at all. They're always present in abyssal samples, but they're never dominant. And then we have tubes. There are lots and lots of tubes in Meiofaunal samples from the CCZ. You will always see tubes. They're really very difficult. They often fragment. Sometimes the tubes are segmented, so it's difficult to know whether to draw the line between these segmented tubes and the homicenids. And another very difficult group are the spheres. You will find lots and lots of spherical agglutinated tests in samples from the CCZ. Sometimes they have a smooth surface, sometimes they're dotted with radial areas. And then we have other things that we just don't know what to do with. They're monothelamids of some sort, all sorts of strange shapes, often with sticomata. Some of these may be Camochiaceans, but most of them we just don't know what they are. Now these are some data from Nina Okawara's PhD thesis from the Kaplan Central site. So this was based on very small syringe sub-core. So this is a 20 ml syringe with the end cut off to make a kind of cora. And she looked at tiny, very small samples, 3.5 mils. So this is a 32 micron fraction. So this is one of the very few studies that's used the 32 micron size fraction. And she found that 77% of the forams were monothelamids, so allogromids, secaminids, and spheres, and the rest were multi-chambered taxa. So the monothelamids were dominant. In the Abyssline project, working mainly in the UK, one area, we've looked at megacore samples at 150 micron fraction. So a much coarser sieve fraction, but we have the same distribution of specimens among taxa. So the monothelamids represent 83% of the assemblage, and the multi-chambered forms represent only 16 or 17% of the assemblage. So again, the monothelamids dominate the individuals, and they also dominate the species. So, again, 83% of the species were monothelamids. Now, there are three problems with monothelamids. The first problem is fragmentation of very delicate, delicate agglutinated specimens. So this is a particular problem with tubular species and Camochiaceans. And these can generate very large numbers of fragments. So this is a tubular form which is broken into fragments, but this is a Camochiacean, this is probably one specimen, I suspect. But it's just very delicate, and it's just been blown apart into lots of tiny fragments. So, what do you do with these fragments? Well, I think they're so abundant that you feel you have to do something with them. But we count them, but we treat them separately from the complete individuals. The second problem is distinguishing live from dead specimens using rose Bengal staining. Now this is a routine operation in foram studies. Foram workers always stain modern samples with rose Bengal in order to differentiate specimens which were live when collected from those which were dead. And this is relatively straightforward with calcareous forams. You can see the stain quite well. But it's much less obvious in the case of some of these monothelamids which have sticomata, but they have very sparse cytoplasm. So there's not very much cytoplasm there to stain. So it's much more difficult in these cases to differentiate live from dead specimens. The third problem is that many species are undescribed. So this is the same sample I showed before. I should have explained, which I didn't, but each of these bars represents a species. And then the abundance of each species is shown on the vertical axis. So we had 169 species, as I mentioned before, and only 20% of these are described. If we look at just the monothelamids, we have 141 species, of which only 3.5% are described. When we look at the multi-chambered species only, 43 species, of which 25, more than half, are described. So the described species are concentrated amongst the multi-chambered taxa. The monothelamids, very few of them are described. So, by geography, a few years ago we published this review of biogeography in deep water forams. So you can have a look at that. It's in the Dropbox folder along with some other literature. And this is a table from that paper, and it shows the distribution of some common deep water species in different oceans and seas. And so agglutinated species are shown in green, the calcareous species in the blue, and the species which we find in our samples from the eastern CCZ are indicated by an asterisk. So, I mean, the message here is that these species really occur everywhere. They're found in all oceans, and even some of them occur in the Red Sea, which is a very poorly studied area. So the idea has grown up that that many deep sea species are cosmopolitan, and there's some support for this from our work in the CCZ. So these are species from or specimens from the UK1 area in the eastern CCZ, and these are specimens from the Porcupine Abyssal Plain in the northeast Atlantic, and they really look very similar, and so based on the morphology you would place them in the same species. These are all agglutinated, these are formicinids, and this is a sort of single-chambered thing which is similar to the formicinids. It's the same story with the calcareous forams. Epistominella exigua, a very well-known deep water foram, occurs in the UK1 area and in the Porcupine Abyssal Plain. Poglindina elegans, another very well-known deep water species, looks very similar in these two areas. And in the case of Epistominella exigua, there's a lot of genetic sequences which have been done in Jan's lab, and these are some part of a tree from a paper by Lecroc et al. Showing very little divergence between these sequences from different oceans. So WED is from the Weddell Sea, ARK is from the Arctic, ATL is from the Atlantic, and P is from the Pacific. So, genetically, this is very similar in these, widely separated parts of the ocean. So, the thing is that, cosmopolitan species are usually described, and they're usually common. Whereas many of the species which are undescribed are represented by only one or two specimens. So again, I've used this graph here. So 169 species, 80% are undescribed, and more than 60% are represented by one or two specimens. So more than 60% are rare. So this is a problem. These are some examples of monothallamous forams that I've only ever seen in the UK1 area. So we can say that some morphospecies found in the CCZ are widely distributed across the world's oceans, and this applies to many well-known multi-chambered species. Other species, mainly undescribed, are known only from the Pacific or CCZ samples, and they may be endemic to these regions. But the problem is that most of these species are rare. So the problem is that the combination of rare species and the very small proportion of the ocean floor that has ever been sampled makes it very difficult to distinguish endemic distributions from undersampling. This is a point Gordon made, and it's a point, I guess, that applies to many deep sea taxa. So ecology. And I just want to say a few words about the responses of abyssal forams to food inputs to the seafloor and their consumption and processing of organic carbon. Basically, you can recognize a number of different trophic strategies amongst abyssal forams. So some species are, they feed on algal material, they're sort of herbivores. Some are opportunistic, they'll feed on sediment or they'll feed on fresh organic material, sort of opportunistic species, and then others are deposit feeders. This is a very nice cartoon from Peter Namaki at Janset, which I borrowed from him. So we've got these three sort of basic types of trophic strategy among abyssal forams. It's been known for some decades now that some forams respond strongly with population growth to inputs of fresh organic matter called phytodetritus, derived from the spring bloom. And so phytodetritus is fresh, relatively fresh organic material which forms in the surface layers of the ocean, settles down to the seafloor, and can form a deposit on the seafloor, which you can catch in core samples. And this is some data, from a couple of decades ago. So May 1998, this was before the spring bloom. July 1998, after the spring bloom. And there's a big increase in the abundance of the talids, these are the calcareous forams, and a much more modest increase in the abundance of monothalamids. So what's happening is that this is phytodetritus in the northeast Atlantic, and phytodetritus is something that David Billett discovered a long time ago. What happens is that this is the seafloor in May, before the spring bloom, and then in July, after the spring bloom, and you've got this deposit of phytodetritus on the seafloor. This is the same area, this is the same mound here. And you can catch this phytodetritus in core samples, and it forms lumps, or lumpy deposits on the surface. If you look inside these lumps, you find forams living inside this material. And the forams always have green cytoplasm, so that they've been feeding on fresh algal cells, and you can actually see the algal cells which have come from the phytodetritus. And this is sort of, consistent with experiments using C13 labeled diatoms as traces of carbonate uptake. So these are some data, some experimental data from Station N, which I mentioned before, on the Californian margin. And the thing is that, in terms of abundance, monothalamids made 57% of the assemblage, calcareous species only 10%. So monothalamids were much more abundant. In terms of biomass, they were similar, but in terms of carbon uptake, monothalamids took up a tiny, tiny proportion of the labeled carbon, whereas the calcareous species took up 42%. So the calcareous species were much more active than the monothalamids in taking up this labeled carbon. So what we can say is that most calcareous species and some multi-chambered agglutinated species have a relatively large cell body in relation to test size. They have green cytoplasm, so they're herbivores which feed on fresh organic material. They undergo population fluctuations in response to food inputs, and we can infer from this that they have high metabolic rates and sort of high levels of activity. I think that's a fair, inference. The relative scarcity of calcareous forams in the CC state probably reflects the low food input, as well as the proximity to the CCD. So they need a relatively large amount of food. On the other hand, the monothalamids are characterized by test contents consisting of stichometer and relatively sparse cytoplasm. They appear to be decoupled from inputs of fresh organic matter. They're probably deposit feeders, possibly feeding mainly on bacteria, and we can infer, and there is some evidence for this, that they have low metabolic rates and low activity levels. So my sort of final thought, I'm coming to the end, and probably, okay. Multi-chambered species are easier to work with than monothalamids, as well as being the most ecologically active component of the fauna. So it could be argued that we should focus on these groups, because these are the ecologically important ones. I think this might be a good starting point. If the monothalamids are too difficult, it might be logical to concentrate on the multi-chambered species, because these are easier and they're more important ecologically. But you will have to bear in mind that you're ignoring 90% of the biodiversity. I think a better understanding of faunal biodiversity and biogeographic patterns will come from a combination of morphological analysis and next generation sequencing. And I think this twin track approach is the best way forward. I've just got a couple of other slides. I'm going to skip over this one because I'll talk about this, I think, during the practical session. But last night I sort of realised that I wasn't saying anything about vents, and you know, we're supposed to consider vents as well as nodule areas. And there's really not very much known about forearms from vents, although Gordon mentioned some data this morning. But this was an interesting study by Cindy Van Dover in 1988 on the East Pacific Rise and Galapagos Spreading Centre, and she put down some settlement plates. I think these were wood, and I think they were slate. I think they were mainly slate. And she found that in one particular site in the East Pacific Rise, that there was a foraminiferan species which densely encrusted the at least one of these plates. There's a second one which I don't know very much about, but this foram species 1 was very abundant. Also ciliates. There are a lot of folliculinid ciliates, which is interesting. Now this is foraminiferous species 1. It was described as Abysotherma pacifica, and it's a multi-chambered, agglutinated, tricaminid, and it's a typical attached foram. So you are likely to find attached forams in the vicinity of vents. And then there are these folliculinid ciliates, and it's just worth mentioning this because this is from these were found by Van Dover, but there's this interesting paper by Corus et al. Published a few years ago, where they found huge quantities, very dense blue mats around vents on the Juan de Fuca Ridge. There must have been millions and millions of these ciliates, and the ciliates live inside these organic tubes. So I thought that was just worth mentioning as a final slide. So thank you to the ISA for funding me and inviting me, and thank you to Anne and your team, and to Gordon as well, of course. Thanks.
Have you got questions?
Yeah, I have actually a comment for you to put in our little tag. These follicular ciliates, you can find them in the Mexican EEZ.
Yeah.
In the Sonora margin.
Yeah.
We found them there associated to the seeps.
Really, yeah. Well, that's interesting, yeah. I mean, there's very little known about non-foram protozoans in the deep sea, but these things seem to be extremely.
We call them blue mats.
Yeah, well, they did as well, these, this other paper.
And we have some samples in case you would like to check them.
Yeah, well, I don't know anything about ciliates.
We are sharing them, we share them with you for now.
Yeah.
Indeed, thank you very much for that very interesting and informative presentation. I have this small problem, I can say. Basically, recognizing foraminifera in Meiofauna. I don't know how many will agree with me or not, but what is the percentage of live forams versus dead we usually get? Because Meiofauna we consider only the live forms.
Yeah.
Therefore, what could be the percentage of that?
Well, it varies from group to group, and usually the calcareous forams are predominantly dead. They're predominantly empty tests. The same applies to the agglutinated, the multi-chambered agglutinated forams are mainly dead. I mean, we always differentiate the two, and everything I showed really related to stained, rose Bengal stained forms. Among the monothalamids, in most groups of monothalamids, they are predominantly live. You do find some empty monothalamids, but usually you can consider them live.
But majority of them, they may not take the West Bengal, isn't it?
Well, yeah, I mean, you have to use other criteria, really. Perhaps this is something we can talk about more on Wednesday when we have the practical session. But the stichometer, these waste pellets, in dead specimens, they break down into a kind of powder. So the test is full of just grey sediment, basically. So that's one criteria. So the freshness of the stichometer. And they do stain as well. Some monothelamids, some monothelamids which contain stichometer stain fairly well. Others don't stain so well. But so it's a combination of stain and the freshness of the stichometer.
That is a major problem when we talk about the percentage of different Meiofauna groups.
Yeah.
The rheumatoids are dominant one, or the hepaticoids and forams usually are very dominant, but it doesn't form that.
Well, you should
percentage.
Yeah, I mean, you should always differentiate between West Bengal stained and dead. So all the ecological stuff is always based on live, quote unquote, forms.
One more, just there is a lot of literature available, but not much have been talked about is the role of forams in nodal formation itself. Would you like to say that?
Sorry, in
Role of forams in nodal formation or binding.
nodule formation. Well, I mean, if you look at nodules, they're covered in forams or in something that we think may be forams. I'm not sure that all the things that we find on nodules are forams, but certainly some of them are. Whether they have anything to do with nodule formation, I don't know. I know Franz Riemann, some years ago, he suggested that they accumulated manganese and iron because he found in the stichometer, he found a lot of iron in the stichometer. And he suggested that they might be implicated in some way in depositing iron and manganese onto the nodule surface. But I don't think they are. I mean, maybe bacteria are.
So what with the Anoxys basin, you found lots of forms in Seabed there? For example, in the Mediterranean Sea and the Black Sea, you have a lot of areas where there are no oxygen, not free oxygen.
Well, there's a lot of ciliates in as I was saying last night, we find forams down to about, I think, 250 meters was the deepest. It was very close to the sulfidic boundary. Of course, the boundary of the sulfidic zone isn't static, and it undulates. I think sort of tidal, there are tidal undulations in it. So maybe sometimes they're exposed to sulfidic conditions, and sometimes they are exposed to some oxygen. So, but I think that was the deepest. I haven't. I mean, I've seen samples from Nelly Segeva from the deep depths. So sort of, she's looked at samples from 2,000 meters and found some very strange things in those samples. But I don't think she's found any forams from the bottom of the Black Sea. But they're all around about the zone, the top of the sulfide. So
Yes.
Maybe you mentioned, and it was that half a minute that I was not paying attention, but do you have an idea how many of the species that are growing on the nodules are exclusively associated with nodules? Can we speak of a true nodule fauna?
Yeah, I didn't really say anything about the nodules because I wasn't sure whether they were Meiofauna. But that's a whole. We could have a workshop on nodule encrusting faunas. Some of them, well, you get calcareous species, which you also find in the sediment. And there are some multi-chambered agglutinated species which are also found in the sediment, but they're usually a minor component. And Saccharisa, these yellow tubes, these orange tubes which you see everywhere on the nodules, they also occur in the sediment.
Okay.
And that's a well-known species.
Okay.
And it was first described, from soft sediment.
Okay.
But the others, I think the mat-like formations, I don't think, they're exclusively on the nodules. You also find other tubes like rhizaminea. There are a lot of rhizaminea tubes on the nodules, and you find those in the sediment, just free in the sediment. So I think there's certainly some overlap, but there is a component of the nodule fauna which is exclusively living on the nodules.
Mm-hmm. But they're not within the Meiofauna that you.
They would be macrofauna, I guess. Yeah, and in the Mullin, there was very nice work by Laura Mulliner in the 80s, and so she compared the densities on the nodules with the densities of macrofauna. She defined them as macrofauna,
Mm-hmm.
which I think is probably right.
Somebody told me, but I don't remember whom, that some forams are like hermit crabs, so that they just take a shell and eat it.
Lots of forams like hermit crabs. So there's a paper by Orly and myself.
It was you, maybe. Maybe it was you.
Yeah, and so the radiolarians, so they live inside radiolarians.
Yeah.
There's lots and lots of them inside radiolarians. Maybe I should have. I didn't want to scare people too much, but often you see the radiolarians are stained, and when you look at them carefully, you can see agglutinated spheres and organic spheres, and you can see tubes, and you can see Chimochiacean-like things inside the radiolarians, and there's lots and lots of them. They're very abundant. And so Orly made a special study of these, and that was published in Deep Sea Research this year. So it's something that forams like doing. They like going inside things. And in the Atlantic, they go inside planktonic forams. Lots and lots of them inside planktonic forams.
Just two questions. First of all, just for clarification, you already mentioned in the samples, very difficult to differentiate or to identify the living cells of the living organisms over there, because some of them cannot stand very well, right? But what's the rough, percentage of the ones after life?
I think most of them are alive.
Most of them are alive.
Yeah. Okay. I think most of the monothalamints are alive.
Okay.
Not all of them. You can never tell if they're alive. They're dead because they're informally. But
when you say the most of them, 70% or 80%, or rough?
Yeah, at least 80%.
Okay. So, second is the practical issues. Based on your presentation, most of those, the organisms not very well established taxa, right? It's not established. So undescribed species.
Yeah.
So what do we supposed to do with them when we try to set up a database for the EIA or what?
Well, I think there should be these categories.
Right.
So this is the scheme.
Yeah, this one's.
So these are the groupings that we use. Maybe I didn't explain this very well, but this is our system. This is our classification system. It's just a pragmatic, morphology-based thing. You just have to do.
So we really can't tell the species A to the species B is really different or the same, but morphology looks different, right?
They look different. More, yeah, I think it's not so difficult to distinguish between species. Although there's some variability. There can be quite a lot of variability within the species. It does take some experience but generally, I mean, you know, you can recognise, you know, like these are very distinctive. You know, these are all pretty distinctive, and you know, you can look at them and you can say that's different from that.
So this group is relatively easy.
Yeah.
The other group looks.
These are relatively easy.
This.
These are easy. Tube is quite difficult, but again, you can distinguish them. I mean, in some ways, it's good because there's no, you know, there's not very much taxonomy, not very much established taxonomy. So you can devise your own scheme.
Yeah, basically it's a practical problem.
Yeah.
For example, like this, the tubes over there, this is from the broken down from one individual, or from the many individuals. When you want to quantify them, you're running into troubles, right?
Yeah.
Because that can affecting how you calculate the biodiversity index and so on.
Well, you have to keep fragments separate from complete individuals. Don't mix them up.
But problem is if you can recognize that the intact individual is just a part of it.
That can be tricky.
Simon and David.
Yeah.
I'm sure that I've dealt with this query in projects in the past, but how does those all work, or what does it do to nucleic acids?
I don't think it does anything to nucleic acids. It's a protein stain.
So you can process samples from a molecular genetic perspective?
I don't know.
Yeah. Yes.
No problem. Yeah. Sorry.
It doesn't work.
Oh, that's not working.
No. But it's more a question of it's ethanol sample usually fixed, and format doesn't work when they're fixing ethanol. So this is more problem with ethanol than elsewhere.
Rosemary ethanol, no.
Yeah, I don't know, but ethanol, unfortunately, is not.
DNA cannot be preserved.
You cannot. You have to fix them and earn a later. And that will happen.
Just like a song.
True.
Yeah, Jan has questions.
It's David first, then Jan. Andy, how many are there of you in the world? And how long does it take to train somebody? And I really appreciate your effort to try and simplify the world for the contractors. And it would be useful maybe for nematodes and for copepods, maybe also thought of ways of trying to do something that is practical with these species. It was to do with your idea about calcareous versus monothalamids, and you said that one was more active than the other, but I think the monothalamids is much more abundant than the calcareous, so collectively they're going to be as important. And therefore I was wondering whether your suggestion of using just the calcareous ones really was likely to be a run-up or not.
Well, I think calcareous plus the multi-chambered agglutinated. So all the multi-chambered taxa, I would suggest. I mean that's, if the monothalamids are too difficult, then one option would be to at least start with the multi-chambered taxa, because they're conventional. Some of them are described. They're easier to deal with. If the monothalamids are just too daunting to work with. The first part of your question about how long does it take? Well, you should ask orally. Maybe, what, year, 2 years?
I've been working with you for 2 years now.
I think there are people who work on them. There's Olga Kamskaya who works particularly with Canochiaceans. And, you know, I mean, there are these splendid Japanese students, and I think Nina Okawara is wanting to get back into research. Yeah, there are people around. It's, you know, they're not that difficult. You know,
Can you tell us?
Yeah. And in some ways, the lack of taxonomic characters is an advantage because you don't have to worry about that.
One is tempted to see you make it up as you go along.
Yeah.
Yeah, you said.
Just a comment. That in fact, there's a solution, of course, that Andy already mentioned, but it seems to be not really discussed very well. So the only solution here is to go through genetics. And we're now working together with Andy on developing a system that will be an automatic system to take piece by piece and extract DNA, sequence it, and then you will get the number of pieces at the end. So I think for the monotymes, we don't really see any other possibility. There's only 3 or 4 persons who knows in the world how to recognize them, especially in the deep sea. And even us, I think, we cannot really say what it is of the species. So if you want to go to the true kind of species list, you have to use genetics. But I will talk about it more.
Yeah.
Is a multi-chambered species, each chamber is 1 year, or how it works?
No, I think it varies. I mean, I think it depends on how much food they have, you know, how quickly they grow. I guess some of the calcareous forams will grow very quickly. You know, if there's phytodetritus on the sea floor, they'll grow up, you know, in a period of a couple of months. So they presumably add changes very quickly. But if there's no food, then they would just be dormant.
Any more questions?
Go for it.
Yeah.